In the previous video, we discussed the primary structure of the molecule of DNA or the nucleic acid, in which we will have alternating a sugar unit with a phosphodiester bond. If we compare that with the protein, we will have that in a protein you have alternating what is called the peptide bond. Let's say we have in this case one peptide bond, two peptide bonds alternating with the alpha carbons, and that sequence of amino acids we say that is going to determine what will be the tertiary structure of the protein, the folding of the protein, and what is the function. In terms of the nucleic acid, we are going to represent the backbone with the phosphate being closer to the nitrogen base number 1, and then we will say that the last nitrogen base by convention will be the one with the free 3' OH. So we have two different backbones. The backbone of the nucleic acid is similar for both DNA and RNA, but in the case of DNA, we need to discuss now what is the history, how the double helix of DNA was discovered. Nucleic acids will be stable. We said before that they are stable because the phosphate units are interacting with histones, but how is that instead of having a single strand like in RNA, the molecule of DNA is a double strand? It's important to remember the convention that the sequence of nitrogen bases is of extreme importance because it is in the sequence of the nitrogen bases what is changing. We say that nucleic acids they will have a repeating pattern. They will have alternating the phosphate units with the sugar, but what changes is the nitrogen bases that are attached to the sugar units. And it's in that sequence of nitrogen bases that we will take that information to make molecules of RNA to determine what will be the sequence of amino acids in a protein. So I want you guys to be very careful when we say that this is a sequence of A, C, T, G without presenting the 5' or the phosphate. It is understood that in this case adenine is the one that has the free 5' phosphate unit, and the guanine in this case is the one that has the free 3' OH. Now we're saying that we have the phosphodiester bond in one strand, but the secondary structure of the molecule of DNA will be a double strand. We will have that these nitrogen bases will be making hydrogen bonds with one another. How is that coming along? So we say yes, the molecule of DNA is a double strand, so we see two backbones that, because of many studies done, is proven not to be like a ladder, but as a twisted ladder, where in the center you will have the nitrogen bases, a purine and a pyrimidine, fitting in a distance very close to each other to make hydrogen bonds interaction. So they will be complementary. So let's talk a little bit about the history of the discovery of the double strand of DNA. The first thing that I want to give credit is to Rosalind Franklin, that in 1951 was carrying out some X-ray crystallography study, and the X-ray diffraction of the molecular DNA was presented by Maurice Wilkins to Watson and Crick. And this picture, this is just a cartoon that I did the drawing, help Watson and Crick to put together molecular models to determine the double strand of DNA. That was the first discovery that was useful. The second one was the Chargaff Radius, in which he did analysis of what was the percentage of each one of the nitrogen bases in molecules of DNA, and he found that in many different species the percentages varies, but it was found that the percentage of adenine was nearly identical to the percentage of thymine, and that the percentage of guanine was nearly identical to the percentage of cytosine. And this one is for human. In the bacteria of E. coli, it is found that the percentage of adenine and thymine is 24%, and the percentage of cytosine and guanine is 26%. If we add all of these together, it's going to make 100. But we also have for salmon, 28% adenine and 28% for thymine. It's always the same. And then for 22% for guanine and for cytosine. So putting together these two information, the percentages of the nitrogen bases and the X-ray crystallography, then Watson and Crick came with a proposal. They actually built using those balls and sticks together, and they discovered that cytosine will always make 3 hydrogen bonds with guanine, and that's what we're showing in here. 3 hydrogen bonds between guanine and cytosine. So you have, and 2 hydrogen bonds with thymine and adenine. Thymine and adenine. And this is always going to be the same. Adenine and thymine, 2 hydrogen bonds. Guanine and cytosine, always 3 hydrogen bonds. And that is of extreme importance, because what that means is that if we have, let's say that this is one part of the ladder, and we have one adenine, thymine, cytosine, guanine, adenine, we know that this is the 5' end, that this is the 3' end, and we have these 2 back bonds. Just because of these discoveries, I will say the 3 discoveries, Franklin, Chargaff, and Watson and Crick, we say that if an adenine is on this side of the ladder, and then we know that this double strand is anti-parallel, on the other side we know that it's going to land a thymine. If we have an adenine on this side, we will have on the other side an adenine. If we have a cytosine on this side, we will have on the other side a guanine, and then in this one we will have a cytosine and then a thymine. This will be the prime end. Of course, these little lines that I have, I'm gonna bubble up in blue, they are representing the glycosidic bond between the nitrogen base, that's what is in blue, and the sugar unit. So we say that yes, this is the sugar unit attached on each case. And then this line is representing the phosphodiester bond between 2 sugar units. But we know that adenine, thymine, 2 hydrogen bonds. 2 more, 3 hydrogen bonds. 3 hydrogen bonds and 2 hydrogen bonds. Now, this information is of extreme importance. How is that adenine and thymine are always making 2 hydrogen bonds and GC, so we call them pairs. So we know that this is happening because of Franklin's study, Chargaff study, and then, I'm sorry, here should be Watson and Crick instead of Chargaff twice, okay? So we know that we have base pairs, we know that we have polynucleotides, we know that we have 2 strands that are coiled around each other, and we are making a ladder. They are anti-parallel. We have that the phosphate units are facing away and they are stabilized by histones, and then these hydrogen bonds are going to stabilize the molecule of DNA. That's one way of stabilization. And the second way of stabilization is by what is called the stacking of the nitrogen bases. Let me say that I have one backbone alone, and I have adenine, and I have thymine, and I have guanine. All of these are kind of overlapping each other, And these are hydrophobic centers, so those nitrogen bases are in the center protecting that information, so there is not going to be any damage. One more thing is that with the twist of the molecule of DNA, we will find what is called the minor groove and the major groove. The minor groove and the major groove are sites where big molecules can bind. As a matter of fact, we know about many chemicals in smoke of cigarettes that can bind, that they can intercalate in between nitrogen bases. So if we have one of those contaminants, let's say we have like anthracene, that can intercalate between those nitrogen bases, that is actually a poison. It can damage the molecule of DNA. But why is this happening that way? We say yes. Now we understand these little dots represent the hydrogen bonding. And the rationale is that cannot be two purines, because if we have two purines, they are going to be way too close, and that's going to be an steric clash. If we have two pyrimidims, they will be too far apart. The molecule of DNA is very symmetric, so we see that the dimensions are very close to 0.3 nanometers, or let's say 30 angstroms. So they are very close to each other. So they have nearly the same distance between the two different base pairs.